Hello everyone, myself Sara Khan from Group 322, and today my topics are generation of the nerve impulse and ionic channels. So, let's start with generation of the nerve impulse. A nerve impulse begins when a neuron receives a chemical stimulus. The nerve impulse travels down the axon membrane as an electrical action potential to the axon terminal. The axon terminal releases neurotransmitters that carry the nerve impulse to the next cell. So it travels from one cell to the other cell. A nerve impulse also known as action potential, is generated when a neuron's membrane potential is changed by chemical signals from a nearby cell. So neurons have a resting membrane potential around minus 60 millivolt. This is due to a layer of negatively charged ions on the outer cell membrane. This separation of charge results in electrical potential, which at rest means not during an action potential when there is no action potential, is 60 MV, more negative in the cell interior. So inside the cell it is negative and outside the cell it is less negative. This potential is generated from the movement of sodium, potassium, and chloride ions into and out of the cell, down their concentration and electrical gradients with the sodium potassium pump. Actively pumping sodium potassium against their concentration and electrical gradient to prevent concentration equilibrium. Potassium concentration inside a neuron is high. So inside a neuron, potassium concentration is high, whereas sodium concentration is low. So potassium are driven out of cell by concentration gradient, while and sodium are driven inside the cell. So when there is it is a flow from high concentration to the lower concentration. Nerve impulses are dependent on the ability of resulting action potential to propagate down the length of the axon without losing amplitude. This process differs in myelinated and unmyelinated axons. So for myelinated and unmyelinated, they are different. The properties of neuroids allow conduction of action potentials along the length of neurons. When a neural impulse reach the end of the neuron, it must be transmitted to a neighboring neuron to communicate with the rest of the body. So it flow from neuron to neuron and then it communicate to our body. This process called synaptic transmission can occur electrically or chemically. So this process is called synaptic transmission. It can be electrically or chemically. So now come to the next topic, ionic channels. So what are ionic channels? Ion channels are specialized proteins in the plasma membrane that provide a passageway through which charged ions can cross the plasma membrane down their electrochemical gradient. So here also there is a electrochemical gradient, so they cross it. The resulting ionic current generated by the movement of charged ions through membrane channels can be measured by patch clamp methods. Ion channels are pore forming membrane. So what are ion channels? Ion channels are pore forming membranes. Proteins, they are a type of proteins that allow ions to pass through the channel pore. Okay. So it is mainly like it allow in and out inside the cell. Their functions, what are their functions? Their functions include establishing a resting membrane potential, shaping action potential, and other electrical signals by gating the flow of ion across the cell membrane. So controlling the flow of ions across secretly and epithelial cells and regulating cell volume. So mainly their function, there are three functions, like they establish a resting membrane potential, shaping action and potential, and other electrical signals. Ion channels. Ion channels are present in the membranes of cells. Ion channels are one of the two classes of ionophoric proteins, the other being ion transporters. The study of ion channels often involves biophysics, electrophysiology, and pharmacology. So, ion channels play a great role in biophysics, electrophysiology, and pharmacology while using techniques including voltage clamp, patch clamp, immunophysicochemistry, X ray crystallography, fluoroscopy, and RT PCR. Their classification as molecules is refers to as channelomics. So, they are classification as when we talk in a molecule level, it is called channelomics. Ion channels are located within the membrane of all excitable cells. So ion channels are located in every cell. Okay, they are located where? Within the membrane and of many intercellular organs. Not only in cells, they are also present in intercellular membranes. They are often described as narrow. What are their properties? They are narrow, water filled tunnels. They are a type of tunnels which are water filled that allow only ions of a certain size. So, obviously, they are ion channels, so they will only allow ions and also of a certain type, of a certain size, and charge to pass through. So, it is obvious that they are ion channels, so they will allow only ions and charge to pass through. This characteristic is called selectively permeability. So we also know selectively permeable membrane. So it is also selectively permeability allow only a particular like ions and charge allows only them to pass. Ion channels are integral membrane proteins. So they are proteins. What type of protein? Integral membrane protein, typically formed as assemblies of several individual proteins. They are formed like a assembly